In this video, we are going to show you how to take water and surface samples to test for bacteria and viruses that could be harmful to your crocodiles, both in their general health as well as skin quality. You can use this type of test to diagnose a condition or to assess the effectiveness or timing of your hygiene regime. There are two ways to do these tests. The first is microbiology and the other is using PCR genetic testing. There are pros and cons to both methods, which we will describe now. Microbiology is the culturing of bacteria as well as yeasts and molds. The microbes must be alive at the time of plating because obviously they won't grow otherwise. With population cultures, often a few different types of nutrient media sources are used, but generally the culturing conditions are quite constant in regards to temperature and aerobic environment. The limitation of this is that not all bacteria grow on the media provided or may have different thermal requirements or be anaerobic. For example, Dermatophilus is an anaerobe, requiring 10 to 15% carbon dioxide to grow, so it is unlikely that they will be detected in a standard lab culture. No viruses can be detected using plating techniques. That is, West Nile virus, pox virus and herpes virus can't be detected using this method and require either cell culture or PCR. If using this method, samples must be kept on ice and received by the lab within 24 hours of sampling. With only 5% of bacteria thought to be culturable, an increasingly popular method is to use genetic testing. Population testing can be done using the 16S gene. The disadvantage is that often the disassociation can only be done to the family level and not the species level. For example, a bacteria might only be distinguished as Dermatophilus species and not Dermatophilus crocodili. The other type of testing is via PCR, which specifically targets a particular bacteria or virus. So there is some versatility in genetic testing depending on what you are trying to achieve. Genetic samples can be stored at room temperature for a short period without affecting the sample integrity, but should be frozen if you are not shipping to the lab in the immediate future. The exception is if you are wanting to test for West Nile virus or similar RNA viruses. RNA degrades quicker than DNA viruses. These should be frozen and shipped without defrosting. To cover all options, we are going to discuss water sampling for microbiology and water sampling for genetics, as well as swab sampling for microbiology and genetics. Before we do that though, think about the questions you are wanting to ask. For example, are you wanting to assess if your cleaning regime is working? Or you can be more specific and ask, is my cleaning regime working to reduce Dermatophilus or pox virus, etc.? Another question might be, I change my water after 3 days. Can I get more days without incurring a problem? More specifically, when does the pox virus load get to cross-contamination levels? You can also just implement monitoring programs to confirm West Nile virus is not present or your staff are doing their jobs effectively. We will start with testing water using microbiology. Always use gloves to prevent contamination from bacteria on your skin, as well as to protect yourself, particularly if you are sampling wastewater. Ensure all cuts on your body are covered, remembering that crocodiles are defecating into pen water, so the risk of fecal coliforms is inherent, so treating every water sample as a biohazard risk is a good precaution. Using a sterile bottle, lower gently into the water at a 45-degree angle. This stops further aeration of the water, which may disrupt the microbial profile before it gets to the lab. Leave an air gap at the top of the bottle. If not, the aerobic bacteria will run out of oxygen and will again alter the reported microbial profile. If there is a possibility of chlorine being present in the sample, let the lab know, as they will need to add a chemical to counter the chlorine before culturing. Label the bottle with the sample ID and fill out the submission paperwork. Put the sample on ice and deliver to the lab within 24 hours. If you are wanting to do genetic testing instead, particularly if you can't get the samples to the lab within 24 hours, this is the method to follow. Instead of using a bottle, we are going to filter a sample of water through filter paper, which traps the microorganisms. There is a special device that we use that can be arranged with the Centre for Crocodile Research. Wearing gloves and using as aseptic techniques as possible, place the filter paper on the platform of the device and seal inside. You can pre-prepare these. Fill the sterile syringe with 50 ml of water, then push the filtration device onto the end of the syringe. Squeeze through as much water as possible. For really dirty samples, you may not be able to get all of the sample through, and that is fine. Just record how much was able to be filtered so that the results can be reported in a standard per litre manner. Using as aseptic techniques as possible, remove the filter paper and store in a sterile tube. Label the tube with the sample ID and fill out the submission paperwork. If sending immediately, ship at room temperature. But if samples are to be batched, then it is best to freeze until shipment. The filtration devices are reusable but require sterilisation prior to reuse. This is simple to do by washing the items in warm soapy water and rinsing well with water. Finally, immediately prior to reuse, rinse everything in 70% ethanol. We can use swabs to obtain microbial profiles from pen surfaces, to define skin problems, as well as understanding contamination points such as cleaning equipment and footwear. Again, if you can get the sample to the lab within 24 hours and are not wanting to culture viruses or dermatophilus, using a gel tube to take the bacterial swab is appropriate. The gel at the bottom of the tube is a nutrient media to keep the bacteria alive until they get to the lab. Open the packaging with gloves on and take the swab out. Do not touch the stem of the swab. Using a twisting motion, swab a 1 centimetre squared area. Then take the lid off the gel tube and carefully insert the swab, trying not to touch the sides of the tube. Make sure the swab inserts into the gel media at the bottom of the tube. Label the tube with the sample ID and fill out the paperwork. Put the sample on ice and deliver to the lab within 24 hours. If trying to detect viruses or less culturable bacteria, then taking a swab for genetics is more appropriate. The method is almost identical for the microbiology swab, except that a dry swab can be used instead. Again, open the packaging with gloves on. Without touching the stem of the swab, using a twisting motion, swab a 1 centimetre squared area. Then carefully replace the swab inside the tube, trying not to touch the sides of the tube. Label the tube with the sample ID and fill out the paperwork. If sending immediately, ship at room temperature, but if the samples are to be batched, then it is best to freeze until shipment. Sampling packs can be ordered from the Centre for Crocodile Research, so just get in touch with us if you need any of this equipment. If you need any further information, please don't hesitate to contact us. We can assist you in developing a strategy to start evaluating your hygiene regimes and understanding your skin quality issues. We can also assist in developing monitoring programs to ensure that your hygiene regimes are still appropriate and your control points are still effective. Remember, these are all aimed at increasing your business's profitability.